Okay, so good afternoon, ladies and gentlemen. Welcome everyone who is watching our next seminar belonging to NeuroEYEscience series. Today is my big pleasure to introduce you Professor Maria Laura Amadio from University of Pavia in Italy, who will talk about potential involvement and target identification of HuR ELAV1 in age-related ocular pathologies. Just for your information, during the seminar, during Professor Amadio's speech, there is a possibility to ask question in live chat. So feel free. We will try to answer all the questions. If you have any questions afterwards, there is an email in the description under the video on YouTube. There is Professor Amadio's email. You can contact her with questions as for discussion in the end of the lecture or after the lecture. The lecture will be available online on YouTube, so feel free also contact afterwards. So, Professor Amadio, the YouTube is yours.
Okay, good afternoon. First, I would like to thank Dr. Adrian Smedowski, that is, first of all, a dear friend, for inviting me. It's a great pleasure to participate in these webinars. As he said, I work at the University of Pavia, in particular the Department of Drug Sciences. University of Pavia is the second most ancient university in Italy. And Pavia is a nice middle-aged city near Milan, rich in art, culture, and history. I started to work on the fascinating ELAV family in 2001, and now my main interest is for the member HuR ELAVL1 in age-related ocular pathologies. The summary of my presentation. I will give you a brief overview of the general context in which the ELAV family acts. Then I will tell you something about the main findings on HuR in 3 widespread ocular pathologies in the elderly, in particular diabetic retinopathy, glaucoma, and age-related macular degeneration. For each of them, my aim was to understand the role of HuR, if it's a good or bad player, and so if it may represent a potential new therapy target and how to pharmacologically modulate it. So, in general, gene expression can be regulated at both transcriptional and post-transcriptional level. Many studies highlighted the discrepancy between the amount of messenger RNAs, the transcriptome, and the corresponding relative protein amount, the proteome. And it seems that around 30% of changes in protein abundance cannot be explained by alteration at the transcriptional level. This emphasizes the importance of post-transcriptional control that is relevant to finally, precisely, timely, locally control gene expression. In particular, post-transcriptional control comprises all the steps of RNA metabolism, such as splicing, polyadenylation of messenger RNA, its transport to the cytoplasm, where it can be degraded, sequestered. So it's important for localization, modulation of stability of a messenger, of translation, are all relevant to precisely affect the amount of a given gene product. So in this context, we can imagine a proscenium where several actors play in concert. In particular, here we have the Greek theater in Syracuse, in the south of Italy. So many factors can affect the fate of a given gene transcript. For example, RNA binding proteins, RBP, and many non-coding RNAs. It's a very complex network called RNA regulon, and it is a highly conserved feature of eukaryotes. Now I will focus on RBPs, in particular on the family of ELAV proteins. ELAV gene was discovered in Drosophila melanogaster. It's RNA binding protein, and its mutation are responsible of abnormal development of the nervous system, mainly the visual system of the fruit fly. And they lead to death within 1 week from birth, from which the acronym. In mammals, there are 4 ELAV proteins, also called HuR proteins. HuR, also named HuA, ELAVL1, is ubiquitously expressed. HuB, C, and D, ELAVL2, 3, and 4, respectively, are expressed selectively at the neuronal level, and they are called neuronal ELAV and ELAV. HuB is also present in gonads. The primary sequence is highly conserved. Here you can see the schematic representation of an ELAV protein that is composed of 3 RNA recognition motifs, RRM1, 2, and 3. In particular, the first 2 are involved in the direct binding to a given transcript. The third one is involved in the binding to the poly tail of the transcript, and it's important for the formation of homo-hetero multimerization, so interaction with other RNA binding proteins. And this domain is separated by the first 2 by a hinge region that is the most variable part of the protein and is relevant for the nucleocytoplasmic shuttling. The post-translational modification of ELAV proteins, such as phosphorylation, is important for the localization and the activity of the protein itself. But how do ELAV act? Following a given stimulus, ELAV protein is activated, and for example, it's phosphorylated, and it can shuttle between nucleus and cytoplasm. It binds to adenine-uridine-rich elements, mainly present in the 3' untranslated region of a given transcript. Although there are other sequences, but these are the most important. And this consensus is quite common. So, ELAV protein can affect all the steps of RNA metabolism, from splicing to translation. However, RNA stability and translation are the most studied processes. We can say that ELAV proteins are main regulator of gene expression, and they are implicated in several physio and pathological contexts. In particular, HuR is a regulator of regulators because it can affect the expression of some RNA binding proteins and many other transcripts encoding proteins factors involved in very different processes, such as cell stress response, cell cycle progression, survival, inflammation, neoangiogenesis, autophagy. So HuR may play even opposite roles depending on the context, the transcript that is bound. That's why it can be considered as a Young's difference. And importantly, in a study performed with Adrian Smedowski, we found that in the rat retina and optic nerve, there is only one member of the ELAV family, HuR. But why focusing on the eye? Despite the discovery of ELAV gene is linked to the visual system, if we search in PubMed, we can find a very small number of studies on ELAV and eye and ELAV and eye disease, representing more or less 3% of the general production on ELAV topic. And some evidence from literature have suggested that HuR may represent a potentially relevant tile in aging and neurodegeneration. Also, loss of homeostasis has been linked to neurodegenerative pathologies, and these may also be the case for the eye, that is considered a window of the brain or a window into the brain. So, considering some of the most common ocular pathologies, diabetic retinopathy, glaucoma, and age-related macular degeneration, we can state that these are priority eye diseases because they are still lacking effective treatments. So this is due also to the poor knowledge and understanding of the molecular and the cellular basis of the diseases, so the ethiopathology. That's why we need to find new therapeutic targets. Briefly, we know that the retina is the most important part of the eye. It is organized in multiple layers. In particular, photoreceptors in the outer part, bipolar cells and retinal ganglion cells constitute the vertical pathway or visual pathway, transducing the light signal into an electric signal that is transferred to the brain through the axons of ganglion cells that constitute the optic nerve. I want to start with HuR, ELAVL1, let's call it HuR simply, in diabetic retinopathy, because I dedicated more than 12 years of my efforts to this study that was made in collaboration with the University of Catania, mainly with Professor Claudio Duccolo and team, and of course Professor Filippo Drago. So diabetic retinopathy is a common complication of diabetes, and it is characterized by alterations in both neural and vascular level of the retina, leading to visual defects and, in the worst cases, blindness. Vascular endothelial growth factor, VEGF, plays a main role, and its signaling is upregulated. Indeed, anti-VEGF drugs are commonly used and are effective, although with several limitations. Some key concepts. VEGF messenger RNA is a target of HuR. In different in vitro cellular models, such as pericytes, endothelial cells in both mono or co-cultures, we demonstrated that activation of protein kinase C leads to upregulation of HuR, its phosphorylation, its binding to VEGF messenger RNA. HuR is activated and binds more efficiently to ribonucleoprotein complexes, to cytoskeleton, where messenger RNA of VEGF is translated. This was also demonstrated by us in collaboration with the group of Professor Miriam Grospe, very famous in the ELAV world, that HuR is very important for the translation of messenger RNA of VEGF. We demonstrated, we confirmed this evidence in HeLa cells in hypoxic conditions. But coming back to our context, the eye context, we demonstrated that PKC in particular beta isoform HuR VEGF pathway, is present also in the retina, and that following diabetes induction in the animals, this pathway is activated. In particular, PKC beta phosphorylase activates HuR, that in turn binds the messenger RNA of VEGF, increasing its translation and thus contributing to the upregulation of VEGF that is important for pathological alteration in the retina. We also found that in vivo treatment with a selective PKC beta inhibitor can blunt, can counteract the activation of this pathway. Of course, considering that PKC is a wide spectrum kinase and its inhibition may lead to some undesired specific effects, more recently we better tailored our strategy, focusing, targeting directly HuR, in particular by siRNA. So in this in vivo study, we used the same diabetic model as a previous study. And the animals were injected with the streptozotocin inducing diabetes, or vehicle, of course, in the case of control animals. 10 days after, the animals received intravitreous injection of siRNA against HuR, either naked or in lipid nanocarriers. We tested different nanocarriers with different composition, but now I will focus only on lipoplex 1, the most interesting. 2 days after, the animals were sacrificed and we analyzed the retina, finding that, confirming that HuR and VEGF protein levels are upregulated in the retina of diabetic animals. Both naked and in nanocarrier siRNA against HuR decrease HuR and VEGF, reinstating their normal levels. Also, we considered, we evaluated the morphology of the retina, because diabetic animals present thinner retina, and we found that siRNA in lipoplex 1 improve the morphology of streptozotocin treated animals. So the retina of these animals improves. According to these results, recently Platania and co-authors demonstrated in vitro that molecules disrupting HuR VEGF mRNA complex are useful in conditions characterized by glucose-induced damage, in particular in retinal endothelial cells. So overall, these findings indicate that HuR is upregulated in diabetic retinopathy, It contributes to VEGF upregulation, and it can be efficiently modulated. We demonstrated it in vivo by PKC inhibitors and siRNA against HuR. So this suggests that HuR may represent a new potential therapy target, and targeting HuR may help in pathologies featured by VEGF upregulation, such as diabetic retinopathies or other diseases. Moving to the second pathology, ELAV in glaucoma, I want to say first that this is made in collaboration with Adrian Smedowski and the team. Indeed, this field of research started in 2015 when we met. Glaucoma is an irreversible degenerative pathology involving retinal ganglion cells. So there is a degeneration of these cells and, of course, the optic nerve. And this degeneration leads to sight defects, in particular to tunnel vision. But before these alterations, the pathology, the degeneration proceeds for many years in absence of subjective symptoms. That's why glaucoma is nicknamed the silent thief of sight. And we do not know yet the reason, the alterations underlying this pathology, why retinal ganglion cells degenerate. So a possibility is that there is an imbalance between endogenous neuroprotective factors and detrimental factors, leading to an increased susceptibility of RGC to stress, to damage. And in the literature, we found evidence that HuR is important in neuroprotection in the brain. Here are some references. So our hypothesis was that HuR may represent also in the eye, in the retinal ganglion cell, a neuroprotective factor in virtue of its binding to messenger RNA encoding factors, proteins relevant in the stress response. The first mention of HuR potential involvement in glaucoma comes from the group of Mochizuki and Russell. They found in vitro that following a given stress, hydrogen peroxide, HuR is activated. It translocates from the nucleus to the cytoplasm when it is upregulated in human trabecular meshwork cells that are relevant for the flow of aqueous humor out of the eye. And indeed, increase in the intraocular pressure is among the risk factors for glaucoma. However, even an optimal control of IOP sometimes does not allow to control the pathology and to slow down the degeneration of retinal ganglion cells. So we studied for the first time in vivo HuR involvement in glaucoma. In our experimental model, animals were intraocular intracameral injected with occlusive polystyrene microbeads on one eye. The other eye was used as a control. These occlusive microbeads lead to a long-lasting, sustained increase of IOP. Different groups of animals were sacrificed at selected times. But here I will focus on the 8 weeks glaucomatous animals, the most interesting, because they are featured by retinal ganglion cell loss, degeneration. Indeed, we found a marked decrease of beta-3 tubulin protein levels. This is a marker of retinal ganglion cells detected in both soma and axon. Indeed, we also found a decrease of beta-3 tubulin in the optic nerve associated with alteration of optic nerve, of axons, demyelination, shrinkage of axoplasm. All evidence indicating this was a glaucoma attack, a strong degeneration of the retinal ganglion cells. First, we analyzed the expression, the pattern of HuR protein in healthy control. Here you see cross sections of the retina and the longitudinal sections of the optic nerve. HuR can be detected in glaucoma, ganglion cell layer and inner nuclear layer. The latter representing the nuclei of bipolar cells, amacrine cells, horizontal cells. In the optic nerve, again in green color, we see that HuR is present in the axoplasm, in the axons of ganglion cells, the optic nerve itself, and also in the glia. You see these small green spots. They are the glia. They are also stained in a red color for GFAP. In 8-week glaucoma rats, first we found, of course, a thinner retina, an impairment in the number of retinal ganglion cells, and an impairment in HuR protein content. This is clear in both GCL and inner nuclear layer. Also, we found a strong impairment of HuR content in the optic nerve, in the axons of retinal ganglion cells. We can appreciate also the alteration of axons themselves. And we found an upregulation of HuR in the glia that is stained most strongly for GFAP in red color. This is an index of reactive gliosis current with chronic damage with a glaucoma attack. We had also the opportunity to consider, to analyze HuR protein expression in human samples, post-mortem human retina of healthy patients and subjects with primary open-angle glaucoma. As in animals, we found HuR in the same layers I mentioned. And look, HuR is diffuse, is present in both nucleus and cytoplasm. Instead, in glaucoma, we see first an impairment in retinal ganglion cells, but important, an altered pattern of HuR. HuR is decreased first, and it is like sequestered in the nucleus. It disappears almost from the cytoplasm. And coming back to the animals, we confirmed these findings. Indeed, HuR in green, in red instead beta-3 tubulin, we see again an alteration in beta-3 tubulin in glaucoma animals, and also a decrease of HuR that is within the nucleus. And we confirmed this evidence by self-fractioning, separating the nucleus and the nuclear fraction from cytoplasm. And we confirmed that HuR is strongly decreased in the cytoplasm at the retinal level, likely the cytoplasm of retinal ganglion cells. And beside this alteration, we found a downregulation of HSP70, that is a stress response factor whose messenger RNA is a target of HuR. Conversely, we found an increase of P53 in the nucleus at the retinal level of glaucoma animals. P53 is a pro-apoptotic protein whose messenger RNA is again a target of HuR. So our hypothesis is that HuR alteration in level and localization can lead to an alteration of RNA homeostasis in favor of pro-apoptotic outcomes. So if this is our hypothesis, if HuR is sequestered, it cannot support the expression at post-transcriptional level, of course, of genes, proteins relevant for the stress response. So we think that its impairment is an example of the deficit in a neuroprotective factor of retinal ganglion cells, having a role in glaucoma. Last but not least, HuR in age-related macular degeneration. a study carried in collaboration with the University of Eastern Finland in Kuopio with Professor Kai Karaniranta team. AMD affects the central part of the retina, the macula, and it is in two forms, dry and wet. The former is very common and completely lacks therapies. One of the earliest events in both dry and wet AMD is the degeneration of retinal pigment epithelium in the outer retina. This monolayer is very important for the homeostasis of photoreceptors and in general for the retina. Compared to control healthy RPE, AMD RPE are featured by increased oxidative stress and many other alterations. And this leads to degeneration of RPE followed by death of photoreceptors. This is responsible of sight alteration and blindness in the worst cases. Among alterations in the AMD RPE, we can find a dysregulation in the protein clearance. So, AMD RPE present intra and extracellular deposits. In particular, the latter named drusen's and are a hallmark of AMD. This accumulation of damaged unfolded proteins is due to an insufficient clearance, so due to alterations in autophagy and proteasome clearance. And inhibition of autophagy was demonstrated by an upregulation, an accumulation of P62 or sequestosome 1 in pink color in this image from our publication. P62 is a stress response factor, a cargo receptor binding to ubiquitinated proteins and organelles and involved in protein clearance, linking proteasome autophagy, and is a marker of autophagy because P62 itself is degraded by autophagy flux. So its accumulation means that autophagy is impaired in this context. Important for this context is our findings that P62 mRNA is a target of HuR. We demonstrated this by different techniques, in particular by alpha screen technology and RNA immunoprecipitation coupled with real-time PCR. These binding experiments were performed at the University of Trento in collaboration with Professor Alessandro Provenzano and the team. We found in Pavia, in vitro, that HuR is necessary for the upregulation of P62 induced by proteasome inhibitor, MG132. We demonstrated that in RP19 cells, the treatment, the long-lasting 24 hours treatment with this reagent leads to an increase of protein aggregates and RP cell death. And the treatment of HuR activates autophagy, decreases protein aggregates with post-survival outcomes, so an increase in the cell viability. So we wanted to study the early events in this treatment, because we know that autophagy process occurs in two phases. At the beginning, we need to upregulate all those factors necessary to efficiently activate autophagy. And later, the autophagy is efficiently active, and so we find a decrease in the markers of autophagy itself. But what happens in the early phase, and is HuR involved in this early phase, so important to trigger an efficient autophagy? Only some results, again, selected evidence. We found that following the co-stimulus, the pro-autophagy stimulus, early translocation of HuR from the nucleus to the cytoplasm occurs. HuR is phosphorylated and it binds to p62 messenger RNA that is loaded to polysomes that is efficiently translated. And again, this was made in University of Trento, and we demonstrated that de novo protein synthesis of p62 occurs because when we use puromycin, an inhibitor of protein synthesis, we stop, we cannot see the upregulation of p62. We studied the involvement of different kinases. We selected some kinases according to their well-known involvement in HuR phosphorylation, translocation, or in virtue of the involvement in stress response. For example, we found that JNK may be a new kinase affecting HuR activity, because inhibition, pharmacological inhibitors of this kinase block the upregulation of p62 protein, suggesting that this is necessary for HuR activation and upregulation of p62 itself. So, to sum up, we demonstrated that HuR is necessary for p62 expression, and this is the ideal temporal sequela of the events. As I said, at the beginning, the pro-autophagy stimulus is needed to favor HuR activation that in turn contributes to the upregulation of P62 that is necessary to efficiently activate autophagy. Of course, when it's active, it leads to a decrease of P62 because, as I said, it's involved in the clearance of proteins. Sorry. So, HuR, which role? In my opinion, HuR is an endogenous protective factor, important for stress response, also in the retina, where in physiological conditions it exerts a positive effect because it upregulates the expression of genes relevant for a positive stress response. But in pathological conditions such as long-lasting stress or high-intensity stress, a derangement in HuR function content occurs. So there is an abnormal response leading to either decrease of HuR and decrease of its targets, protective factors such as HSP17 glaucoma, or upregulation of HuR and increase of factors with a pathogenic role, such as in diabetic retinopathy and VEGF. In both cases, we may use either HuR activators or inhibitors to reinstate the normal levels of HuR, because this is an important factor, so normal levels. Of course, we are aware that the context is quite complex because HuR has a pleiotropic role. It binds to many transcripts, so with, we said, opposite roles. Also, the timing may be relevant. So not only the pathological context, but also probably the phase, the stage of a given pathology. Also, we have to remember that other factors regulate RNA. So the picture is complex. We now know that the majority, around 98% of non-coding RNA has a role. For example, it can regulate post-transcriptional gene expression, microRNA, circular RNAs, and so on. It may affect even protein function and even DNA. So we have to consider all these aspects. And to conclude, we can conclude that HuR is altered. It may represent a potentially interesting therapy target, but its modulation is challenging. It's feasible, and it may, in the future, improve, be complementary to current therapies. Here are some bibliography I mentioned during the presentation. Here is something I recently published. This is another main field of research supported by the University of Pavia, the study of NRF2 involvement in AMD. I want to thank all the people I mentioned, all of the collaborators, and at the University of Pavia, of course, Professor Stefano Goroni, Alessia Pascale, Simona Collina for the ELAV adventure, Nicoletta Marchesi, all the graduated and undergraduate students whose work is so helpful in the lab. Here is my email for your question. They are welcome. And I want to thank you for your attention and give you a quick test of your site.
Thank you very much, Maria Laura. It was really broad insight in the topic, in the aspect of HuR. So I have just one quick question to you. Do you think that HuR could be kind of interesting or important target for treatment in future?
I hope so. We have to understand the context first, and in some cases we are quite in a good position, for example, for diabetic retinopathy. For some other context, we need to have further results, but I think it's quite promising. I'm dedicating a lot of time and efforts, and so I hope so. I count on it.
Yeah. Thank you very much. So, ladies and gentlemen, this was Professor Maria Laura Amadio from Pavia, from University of Pavia in Italy. So, as I mentioned before, just please feel free to ask your questions via email, which is provided both on the presentation and also in the on the YouTube channel. And please follow our channel because there will be more lectures in future. So feel invited, and thank you very much for participation.
Thank you, and bye.